Using these analogies, I see very close resemblance between genetics and software. For example, I can consider Genome as an operating system which controls organism, human in this case. And this particular operating system contains set of programs which called chromosomes. And these chromosomes, for example, I will take this part, have these stripes, which I can consider as a library is in our software terminology. And these libraries contains a function which are genes. And genes has their own structure. For example, they have exons which are code from our point of view, and introns which is data from our point of view. And only code goes to when this function is called, because when they transform to messenger RNA, that's a function call from our point of view. But what is more interesting, they have this untranslated region. They do not know why they are all there, because they don't do anything, but nevertheless they go to the resulting executional part when this function is called. And I have wild idea that probably these are input parameters or placeholder for input parameters, as well as this part are placeholder for output results. Again, this is a wild idea, but who knows? And so I decided to put everything in one table, and I see very close resemble between genetics and software. If I consider DNA as a sequence of commands and data, and genome as operating system, and chromosome as a executable program, and region is our library now terminology, and gene looks like a function, and we even have this function call. Very close resemblance. That's why I think that this is very right time to create new scientific discipline which would use digital informational technology methods and approaches, I would say IT way of thinking, to explore genetics tasks from informational point of view. And I decided to call it digital genetics. And what it does, first we need to explain somehow patterns of some matrix. And we need to find all biological meanings of these patterns. And we need to define digitally most of genomic terms, entities like chromosome, gene, messenger RNA, join, for example, and so on and so on. And so matrix should be analyzed for comparison of different genomes or chromosome of the same genome, or maybe if it's feasible to compare sequence for the same person on different times in his life. Or we can try to say subtract or add in two genomes one to another. For example, we can add viruses to genome or maybe subtract. And I even can imagine what's possible digital experiments can be done. But what I can see the ultimate goal of digital genetics, this is to define the instruction set and architecture of the biological processor which we call life. This is ultimate goal of digital genetics. Though we have immediate things which we can do right now, we need to create some tools for redigitizing current databases and transform all of them into normal binary hex format. and we need tools for calculating some matrix and for comparison some matrix and presenting the results of this comparison somehow. And we need tools for analyzing patterns and just to find out how many patterns are there and does they mean anything. And probably we need to rethink genetic code table for amino acid production, because this is very strange to have triplets like they have, triplets codons. It doesn't sound right, and probably we should do many more things. And what I would like to say in conclusion, I always say we and them, but from us software IT professionals to them genetics and biologists, I would like to say let us help you to help all of us. And this is the conclusion of my presentation. And you can find all this description in my blog, which is summatrix.blogspot.com. And thank you for listening to me. I am Victor Sumskoy, software enthusiast, and thank you for your attention.